Okay, welcome back, everyone. So thank you for joining us, and hopefully you had a good break. Just briefly before we get started, to introduce the rest of this afternoon session. So we finished our short talks and panel discussion. Thank you very much to all our speakers. So now we have our final plenary for the day, before which we have a wrap-up discussion, which we've invited a few extra participants to join us for that. And then we'll finish with our breakout sessions. But we're really, really excited to have our next plenary speaker. So thank you very much to Professor Neil Shubin for joining us for the next plenary talk. So Professor Shubin is a professor of organismal biology and anatomy, and is really been exploring the mechanisms and evolutionary origin of anatomy and biological systems. So thank you very much, Professor Shubin, for joining you. Hopefully you're able to share your screen.
Let's give it a try. Let me just see if we can get it together.
Fantastic.
Can you see it and hear me?
We can see you and hear you. So thank you so much, and I'll hand over to you. Thank you.
Okay, thank you so much. It's a real delight to be here. I want to thank the organizers for inviting me. And what I wanted to do is just to bring my own perspective to today, to the meeting. And my perspective comes as an evolutionary biologist, as you heard. And I'm motivated by the question, how did the great transitions in evolution happen? How did fish evolve to walk? How did bodies come together from single-celled microorganisms? How did animals evolve to fly? How did animals return to the water like whales from terrestrial creatures? And how did these great transformations, particularly in animal systems, happen? And I approach this from a variety of perspectives. Mostly, paleontology, going out and collecting fossils. We'll talk a little bit about that today. Genetics, to look at the genes that build bodies and how the genetic recipes that build bodies change over time and what that means. And finally, I think of the relationship between locomotion, feeding, and sensation. And we're going to end the talk with that today. So what I thought I'd do is to kind of try to demonstrate why evolution happens in ways you might not even imagine. It's much more multi-textured than the textbook stories that are out there. And I'm going to give you a few of them to show how the course of evolution can not always easily predicted. And then what I want to do is to show you the different lines of data that we use to understand this and what it all means for the kinds of work that you do. Now, I'm going to open up with a quote. And this woman here, Lillian Hellman, she's going to have the quote that's going to sort of define much of my talk today. Lillian Hellman was a famous playwright in the United States. She was a hard-living person, went through lots of bottles of whiskey, lots of cigarettes, lots of husbands. And so she was writing her autobiography. And she, in looking at her life, had a phrase which captures the great transitions in evolution. So I was reading her autobiography for another purpose and saw this quote, and I realized, hey, it's exactly what I'm going to be telling you today. And that is, nothing, of course, ever begins when you think it does. That is, when we look for antecedents in the evolutionary record, whether it's antecedents in locomotor behaviors and feeding behaviors and sensation and so forth, and the ways that animals interact with the world, the physical world, the course is something that you really can't always think about what the origins are, because the origins are often far more complex. And let me give you two examples for that, which are almost textbook, but I think they convey the power of the point. If you think that lungs arose as creatures evolved to walk on land, or arms and legs and so forth, if you think that feathers arose as creatures evolved to fly, you'd be in very good company, but you'd also be entirely wrong. And we've known that for over a century. Lungs arose well before creatures evolved to walk on land, in aquatic creatures living and exploring in aquatic environments in new ways. And the organs were then repurposed, and I'll show that in a second. Arms and legs, as you'll see in a second, actually arose in fish living in water before they ever took their first steps on land. Feathers arose in dinosaurs, not in flying dinosaurs, but in carnivorous dinosaurs. And they were probably used for thermoregulation, or they're used for color patterns, for mating displays, all that stuff, whatever. But the key thing is that nothing, of course, ever begins where you think it does. And when we think of the major inventions in evolution, the major inventions of locomotion and feeding and sensation and so forth, they had a path that's involved lots of repurposing, cooption, duplication, and sorts and complexities like that, which I think are relevant for many of the questions you might be thinking about. So let me just show a couple of examples here. I mentioned the lung examples. Well, we know that if we look at the origin of tetrapods, the origin of limbed animals from fish, this is a event that happened in the middle to late Devonian. We know that from fossils, and I'll talk a little bit about that in a second. But we have relics of these forms alive today, living lungfish actually. And this is a creature, a lungfish from the Shedd Aquarium in Chicago. And they have big fleshy fins, and we can talk about them a little bit later. But they have lungs, as the name implies. They have lungs and gills, and they use both of those organs. So the idea is essentially that the lungfish, kind of like their distant ancestors in the Devonian, had both lungs and gills, and we can see that in the fossil record. What they do is they use the gills when the oxygen content of water is high. But in these waters that the creatures live in, the concentration can vary throughout the year, such that lungs become an important accessory organ to allow them to live in these waters. So they gulp air. So what typically happens is these creatures come up. This is a classic diagram from like early 1900s, where they come up and take gulps of air, and they have true lungs to do this. So if you look at their internal structure, what they have is a lung that essentially runs the length of the body. That's different from ours, of course. But what's similar is the way it develops. It's outpocketing from the gut tube. So in development, it forms a little outpocketing. But it also has a trachea, it has alveoli, it has surfactants, it has many of the materials, the tissues, and actually, if I'm not showing it, but the genes that form these things are pretty similar to those that form our own lungs. So these are really lungs. And if we look, and again, this is a diagram, I just want to lead it off as this example, which kind of nothing ever begins when you think it does. If you look at a phylogenetic tree of the major groups of fishes, including us, on the right are tetrapods. So all the way on the right there, you can see that lizard-like creature. It's an amphibian. It's we're in that group of tetrapods. Those are birds and mammals and so forth. Lungfish and other fossils are a little deeper in the tree. Sharks are even deeper in the tree, and there's some extinct bony fish even deeper in the tree. If we look at the presence of lungs on this cladogram, on this phylogenetic tree, lungs are primitive. They are deeply primitive, and they arose in aquatic systems, not in terrestrial systems. And in fact, if we sort of map the presence of lungs, there are hundreds of species of air-breathing fish. They'll either breathe with a structure like a lung, or they're vascularized their mouth, or they're vascularized their skin. There are many ways to do it. But air breathing is a very common strategy in these creatures. But it predated the transition from life in water to life on land. And that's my big point here, that the inventions that are so necessary for major revolutions in functional revolutions in evolution and ecological revolutions, that they predate by a long way. And they usually evolve in a different context, and repurposing of the same organ is very important. And so what I want to do is to show this even more example from some of our own work, and to show, kind of give you a sense of how paleontologists actually work, and how we interpret those data, and what those data might mean for the evolution of locomotion and locomotive behaviors. And particularly for the kinds, for thinking about how robotics can inform evolution, can inform an understanding of locomotion and understanding of the great transitions. So what you see here is a textbook diagram, as I showed earlier, of the transition from fish to tetrapod. And it's actually a very simplified cartoon in a textbook. We have lots of fossils that tell us this, but what you see on top is, reduced for simplicity, is a cartoon of a fossil fish from rocks about 390 million years old. And you can see it's a fish. It's much like a lungfish. It's a fossil that is closely related to tetrapods, has a conical head, has fins, but it's clearly a fish. What you see on the bottom is a cartoon of an early tetrapod, early limbed animal. This is a creature from Greenland. It's from rocks that are about 365 million years old. And so I'm just showing these endpoints just to show between 390 million years ago and 365 million years ago, you had a pretty big transition in evolution. You had changes to a head, where you have a conical head going to a flat head. You have an origin of a neck. You could see how the head and the bottom swivels independently of the body, whereas in the fish, it does not swivel independently of the body. It's attached by bones to the shoulder. And of course, you have the transition from fins into limbs, as well as lots of other things going on. But yeah, that sort of captures it. Now, the reason why I'm showing this diagram is that this is an active body of research. We can approach this question by asking, what are the functional transitions that happened, going from different types of swimming to different types of walking? What are the physiological transitions? What are the transitions in the anatomy and the skeleton and so forth? And I want to talk a little bit about, for the next 6 or 7 slides, about how paleontologists actually work, just to give you a sense of how those data are collected, and then to set up an understanding of what happens with locomotion. So if you're a paleontologist and you're asking the question, well, how do I find fossils that bridge this gap between fish and tetrapod? You look for places in the world that have 3 things. You look for places in the world that have rocks of the right age to answer your question. So remember, I told you we're dealing with something in the window of 390 to 365 million years ago. That's the middle to late Devonian, pretty much the period you want to be in. So, and you can find places in the world where there are rocks that are mapped of that age. Different countries have produced maps with geological maps that have that information. The next thing you do is you look for places in the world that have the rocks of the right type to hold the fossils. And in our case, really the big conceptual breakthrough for us was actually a very simple one. And that was coming to the realization that if we could find places in the world that had middle to late Devonian rocks, that is of the right age, that had ancient delta systems inside them, reflected ancient deltas like the Amazon today, what we do is maximize our odds of success. That is, if you look at this delta system here, this is actually a map of what the state of Pennsylvania in the United States looked like 365 million years ago, but it applies to a couple other areas in the world. You have an inland sea to one side, you have rivers and streams that drain from a highlands on the other into estuaries. And so what's nice about this is if you have this environment inside the rocks in a region, you can sample ancient tidal flats, you can sample ancient estuaries, you can sample ancient streams, ancient oxbow lakes, you get the idea. I mean, these creatures are not going to live in the deep ocean. So this kind of setting reflects the diversity of environments where these creatures likely lived. So if you have rocks of the right age of this kind of setting, geological setting, you're maximizing your odds of finding the kinds of creatures that would tell us about how, walking evolved from, terrestrial walking evolved from aquatic swimming. And that's exactly what we were doing. And so we found one of the best places to look with maximizing those 2 variables was in Nunavut territory in Canada. This is in the 1990s. We're working with a team from Philadelphia and Princeton and a few other places, Harvard, and a few other places. And you could see Nunavut territory is the northernmost territory in Canada, and circled in red is Nunavut. It's huge. Now zoom into the main body of the slide. And what you have are the islands of Nunavut territory. You can just see them there. And the reason why I'm showing you this slide is we took this from the geological literature, colorized it a little bit. But what you see circled in red in the main body of the slide is where middle and upper Devonian age rocks formed in ancient delta systems are exposed at the surface. So look at that. You could see the scale bar on the left-hand side. That's 100 kilometers, little bar. That's a pretty vast area where you have rocks of the right age, rocks of the right type, actually exposed to the surface. So if they're not covered by ice or tundra, what you have is the perfect kind of setting for paleontologists to find, relatives of the earliest creatures to walk on land. And that's exactly what we did. We spent 6 years working in this site, 5 of them failures. Fortunately, we had the last one successful. And, we ultimately found of this particular valley, which has what we're looking for. And I'm going to show you this valley again, because it's going to show one of the quirks of evolution in a second. But you can see how the layers of the rock, you see how they're tilted, they're kind of tilted like that. You can see the layers. And, but there was one layer that we found that had ancient fossil skeletons of fish, middle late Devonian age, piled one on top of the other inside the rock. So we had this layer, that we found. And we worked that layer for a period of time. And ultimately, in working that layer, the team was pulling out skeletons of different fish. But the real kind of fish that caught our attention happened here when my colleague found in that layer, you can see 2 people working, This V, do you see where the white arrow is pointing to sort of a V in the rocks? That's an upside down skull. If I showed you in detail, you could see teeth and things like that. So here I had an upside down skull, and also looking at that skull, I knew from the texture of the bones it was fish-like, but I also knew, because seeing the opposite side of this, that it was a flat-headed animal. So I had a flat-headed fish sticking out of me. So that's one. I mean, it was clearly an intermediate. So bring these kinds of things back to the lab. We since have found 20 of them. And you can see what happens after preparing them. Now we do a lot of this with CT scanning, high energy CT. We can zap inside the, image inside the rocks. But back in in the ancient days of 2006, we removed the rocks grain by grain, and you can start to see that particular specimen. You could start to see, you're looking at it in a dorsal or top view, you could see two eye holes or orbits. It looks like almost like a crocodile-shaped skull. A few more months go by, and you can start to see that this head is coming out. You can see it's a flat head with eyes on top, kind of like an early tetrapod, right? You can see shoulder blades behind the head there. Looks like there might even be a neck in this animal. It's clearly a fish because it has fins. And this is what emerged. What you have is a creature known as Tiktaalik rosea. We now have about 20 of these specimens. They range in size from about 1 meter and change long to about 3 meters long. We believe they grew continuously over life. But what you can see is in this particular specimen, which is the one that's about 1 meter and change long, is you could see scales in its back like a fish. They're very fishy. You could see fins in the foreground. Just look at the foreground on the left there, on the right there of the slide. You could see fin rays. You could see shoulder blades. You could see there's a head could swivel independently of the body. There was likely a neck. And it had a flat head with eyes on top. Yet it had a variety of fish-like features, some of which you can see with the scales or the texture of the head. That's very fish-like, the texture of the bones of the head. But much of this is also tetrapod-like. So it's clearly an intermediate. All right, now we can get in business and ask what's locomotion? What's happened? What's the change in locomotion that we see? What does this tell us about movement dynamics, sensation, and so forth? All right, so I had a postdoc in the lab who, in the last 5 years, we have a CT scanner downstairs, was able to put this whole thing in high energy CT scanner. So we now are publishing, actually in a couple of weeks, the 3D reconstruction of the body with all the pieces digitally prepared out. And this is kind of what you have. It's clearly a fish. You could see the fins. You could see the fin rays in blue there. But inside the fin, shown in white, are bones that correspond to our upper arm, forearm, even parts of our wrist. So it has an arm embedded in the fin bones, okay? It has a humerus. It has a radius, an ulna. It has a shoulder, an elbow. It has a wrist inside that. And so, and you can see a large shoulder girdle. Look in the back. You can see this huge pelvic girdle too, with the pelvic fin, with ribs and so forth. So we can put this in a comparative framework. And so this is kind of what I like to show what paleontology can do. So in the top is that fish I've been showing you already. It's known as Eusthenopteron. This is a more detailed diagram of it. And you can see the ribs in pink, the pelvis in blue. Look on the bottom. Those are early tetrapods. These are from rocks 365 million years old. You can see they actually have digits. You can see the fingers and toes on there. They have really big pelvis as compared to Eusthenopteron. Look at the size of the hip. It's really big. Their ribs are bigger, and on and on and on. When you add Tiktaalik, and there are now other creatures like it into the mix here, what you can see is that before you had digited creatures, you already had creatures with fins. They had fin rays with limb bones inside, although they differ from tetrapod limbs and not having true digits. Head shape, beginning to get a big flat head there, getting bigger ribs. Look at the size of that pelvis in Tiktaalik compared to Eusthenopteron. It's of the order of magnitude of an early tetrapod, yet the shape's very different. So we can go by the shapes of these things to ask the question, what are likely and unlikely ways that these appendages move? And where are the antecedents of the kinds of walking behaviors we see in terrestrial fish? Where are the antecedents in some of these aquatic fish as well? And the fact of the matter is, we're already seeing the basis of quadrupedal locomotion in these fish like Tiktaalik. We're already seeing the ability to protract and retract fins, or the limbs, whatever you want to call these things. You're already seeing that these creatures are supporting themselves with appendage-based support. It's clearly a fish. It has both lungs and gills, but these things are probably more tethered to the water than, say, the creatures on the bottom of the diagram. So you're seeing sort of the assembly of not only the bodies, but you're seeing the assembly of the locomotor systems that I think are essential for walking behaviors. And the walking behaviors are initially, a lot of these changes already happen in fish living in aquatic ecosystems. We already know that walking, like lungs, is a very primitive behavior seen in diverse kinds of fish. And they use it in a diverse kinds of ways to walk on complex substrates. They use it to, station hold, support themselves on the bottom as a current's going by. So you have to think about these things as not with optimizing one function. They're optimizing a lifestyle in an ecosystem that requires lots of different functions. And I think that's a key lesson here. That's why, when we think about the transitions, these things are doing lots of different things. And it's not like you have a clear-cut dichotomy between water and land. There are all kinds of intermediates between water and land. Think about the benthos, where animals live on the bottom. Think about the mudflats, or think about the shallows. Think about areas that are intermediate environmentally. And then think about the kinds of adaptations animals use to live in those. Turns out a lot of what we think of terrestrial features, they could have evolved in these intermediate ecosystems, right? Or intermediate parts of the ecosystems, with animals that have using their appendages for a variety of different functions, not just one thing. So it's important to think about the complexity that happens in a natural ecosystem. Now, there's another layer of complexity here that I'll return to this, but there's another layer of complexity. And this, I told you I'd show the Tiktaalik site again. This is the Tiktaalik site. The arrow on the right is the site I just showed you, where that fossil came from. The arrow on the left is another place where another Tiktaalik-like animal, different genus, whole different kind of thing, smaller, all kinds of stuff, was found. And just to give you a sense, we pick up blocks like this. This was work done only a few years ago, 4 years ago, actually right before we were locked out of our offices during the COVID times. But we can scan now inside the blocks, and you can see inside a block like that, which we collected because it had scales on top. Turns out there's an entire fin inside. And for this animal, we now have parts of the head, the jaws, the fins, and on and on and on. And there's a reason why I'm showing you this, because I think it'll show the sort of complexity of evolution, and I think in a very important way. And that is, when we look at this animal, it had a head like Tiktaalik, but had a very, very different kind of fin. And then when we look at that fin, you can see fin rays. They're shown in the orange there. So it's clearly a fin, but it has a humerus, radius, ulna. It has a one bone, two bone kind of tetrapod pattern, limbed pattern. But what's interesting here is this is a much more paddle-like limb. It's a limb that doesn't have the muscle scars, which are for like a big pectoral muscle, which is what we see in Tiktaalik and tetrapods, which would support the body. This is not an appendage that could support the body against a substrate, because there are no muscle scars for that. Furthermore, the joints face distally. They don't face ventrally like you have in Tiktaalik. So it's not a flexed appendage. It's an appendage like a ping pong paddle, Yet it's very Tiktaalik-like. You have the one bone, two bone, you have the same skull. And it gets even, so here's an aquatic thing. And so you'd say, okay, so who cares? It's an aquatic animal. I get that. Aquatic, so it's primitive. But the answer here is, and this is where the surprise is, it's not primitive. It's highly derived. That is, if we look at a ventral view of the humeri, so what you're seeing here is a phylogenetic tree, evolutionary tree, of the different kinds of species that are present in the middle to late Devonian that are relevant here. And this is the ventral view. This is the belly side of the humerus. What you'll see is there's this ridge. Look at Tiktaalik in the center there. You could see a ridge as that dark line going diagonally right across the ventral surface. That's a huge crest for the deltopectoral muscles. They're essentially the ones that control the flexion that supports the body against the substrate. You see that same crest in more tetrapod-like creatures as you go to the right. See Elpistostega, Acanthostega, Ichthyostega. You see that crest there. But guess what? You see that crest also in more primitive creatures. You see it in Eusthenopteron and Panderichthys. They all have that crest. But Kikatania, and this is not based on preservation, this is the real deal, does not have any of those crests for the muscles that are essential to support the body. So what does this tell us? This tells us is that this is a creature that returned to the water, right? That it's lost a number of the terrestrial features, if you will, to support it, that it's actually specializing for living higher up in the water column. So when you think about this, what the story is here, I think it's actually pretty important. That is, what you're seeing is this creature known as Kikatanias in the middle, shown in light green. Tiktaalik on the bottom. These lived in the same place, but you had one specializing for life in the benthos or shallows or even the mudflats by walking around and doing that kind of tetrapod-like stuff. But another one that returned to the water, that lost all that stuff, is actually swimming. So when you think about this, and again, where I want to return to the Lillian Hellman quote, nothing ever begins when you think it does. Remember also that there's no directionality to evolution. We tend to think that evolution was this march of progress from water to land. But what you're really seeing throughout is it's going both ways, right? Animals are using these traits not only to walk, but they're modifying them again to swim in new ways. What that new way is, I don't know, but I know they're swimming. And so it's conveying the complexity and how a structure can have multiple uses, which is really essential because that's what provides the evolvability that I think is so necessary. So when we think about this, with the no sharp dichotomy between land and water, when we think about these ancient ecosystems where animals were evolving to walk on land or walk in the water bottom or re-evolving to swim in new ways, what we're really thinking about is that the origin of features has long antecedents. And those antecedents, that history really involves multiple changes in function, repurposing ancient organs to use them in new ways for whole new activities. So when we think about innovation and evolution, this is a list I'm going to build throughout the talk. Repurposing, a change in function, is a very important thing. And when we think about the changes in function and evolution, we're really thinking of the shift from swimming to walking in water. And then what happens as you transform from walking in water to walking strictly on land. So, but you're using the same features in novel ways. It's not, evolution doesn't always involve, origins of new structures or origins of new genes. It often involves using old stuff in new ways. In fact, it mostly does. And that's what opens up new trajectories. Now, there's another principle I'm going to lay on here, and we're going to go and take this in a genetic level because I think it's important to understand how things come about, their ontogenesis. And that is cooption and redeployment. That is redeploying structures in new places or co-opting ancient structures, functions, genes, and so forth to do new things. So this is all how you build new stuff, right? It's not always that you have a new structure appears. That's kind of what we naively think. It's not the case at all. You're using old stuff in new ways. Repurposing is one way, co-option and redeployment is another. Let me give you a couple examples of that. And this is more developmental, but I think it outlines a really, I think, important and kind of cool point. All right, so you think about our limbs, right? Remember, I've been talking about that so far. One bone, two bones, wrists and ankles and toes and so forth. Developmental biologists actually recognize three segments to the appendage. That the one bone, the two bones, and then the wrist bones and digits. They're given the name stylopod, zygapod, and autopod. Now, as a paleontologist, I can look at this and see how it was assembled, over time. And you can see, that one bone, two bone, wrist and ankle bone thing, digits came about in fossils in the Devonian. You can see that with the earliest digited creature here. By the way, this is on display at the museum in Cambridge. And you look at Tiktaalik, which creature I just showed you, it has those three segments too, but it's still pretty different. I mean, you have the one bone, you have the two bones, you have those terminal bones, but they're not really patterned like digits. And furthermore, they're sitting in a sandwich of fin rays. All right, so what's happening here? How does this all come about? And how do we use living creatures to tell us something about that? Because there's a living creature in the bottom. That's kind of one of the creatures we use for developmental biology. So now we're going to drill into a little bit of genetics for the next 6 slides, but it's an important point because it shows the importance of cooption. And I think it's a really important point. All right, so here's a mouse limb bud. This is a mouse a few hours after conception. The limb bud begins as a little outpocketing of the body. And what the researchers did here is they labeled a gene called a Hox gene, particular kind of gene, and it's labeled in purple where it's active. So this gene, this Hox gene, is turned on in the terminal or distal part of a mouse limb bud. Turns out that part of the mouse limb bud is the part that's going to form the wrist and digits, such that if you trace the cells that had the activity of this gene all the way to the adult stages, it almost perfectly marks the wrist and digit area. You can see that in green. Okay, so it's a marker for the wrist and digit. And if you make a mouse that lacks these genes, what are you missing? You're missing that terminal part altogether. So think about this for a second. These Hox genes, there's a couple of them. They are active in the terminal end of the mouse appendage at the time when digits are being specified. such that when you trace the cells that had activity of these genes in light green here, you'll see it kind of marks the wrist and digits. There's some expression, some activity in the tendon there. I don't know what that is, but it's mostly marks the wrist and digits. And then if you make a mouse that is lacking those genes, guess what you're missing? You're missing the wrist and digits. So this is a perfect marker. It's necessary, but not sufficient, but it's necessary for the formation of wrists and digits. Well, guess what? My lab spent years looking at this. Fish have these too. Check this out. So here's a zebrafish. Look, so marked in purple there is where these Hox genes are expressed in the terminal end of a zebrafish fin. And guess what happens? If you trace the places, the cells that had activity, it's the terminal end. This isn't a fish fin. It's marking all the fin rays. And if we make a fish, use CRISPR-Cas, genome editing, to knock these genes out, we're missing most of the fin rays. So what you have is the antecedents of the genes that are necessary for the formation of our wrists and digits. They're already present in fish. And guess what they're doing? They're making the terminal end of a fish fin. So you can compare like a human on the left to a zebrafish on the right. Those bones don't look very similar, but when you compare them at a genetic and cellular level, you can compare the terminal end. All right, that's cool. And it's kind of reinforcing the point I showed you before, right? That nothing ever begins when you think it does, that you can trace the antecedents for many of our structures and genes actually very deep in time, doing different, similar but different stuff, right? Same but different. All right, now here's where it gets interesting. These genes, these Hox genes, were originally discovered in flies, where whole body segments were duplicated, or an antenna, a leg would form where an antenna was. So these are genes that controlled what organs would form where in a fly body. They were discovered in the mid-late 1980s. Such that when you look at the genes that build the basic architecture that specify where organs are in a fly, they're present, versions of them are present in our own body, specifying the identity of our vertebral column, why our limbs or front limbs are in one place and our hind limbs are another. What I'm saying is that these genes, which were in flies, originally building the body plan of flies, are building our own body plan, but they've also been co-opted not only to build our body plan, but like a subroutine of them has been co-opted to make our appendages. So those genes I showed you are genes that build, originally genes that build the whole body of them, specified the body plan of flies, and versions specified the body plan of humans, but they've been co-opted to form limbs. They've also been co-opted to form genitalia, interestingly. So, and when you look at these genes, here's another principle. It's the principle of duplication and divergence. If you look at our closest worm-like ancestors, the ones that are alive today, they have one set of these genes on a single chromosome, just like flies do. That's what you see there in color. Okay, and you're seeing Amphioxus, which is one of these worms. If you look at the phylogenetic tree, what you see is the duplication of these genes over time. So when you go from Amphioxus to jawless fish, you have a duplication. You now have 2 copies of these on 2 different chromosomes. When you go to sharks, you have 4 copies of these, just like mouse. And when you get to some fish, you have even duplications even more. So what you're seeing is an increase in complexity of the genetic pathways of development. One of the prime ways that happens is, number one, the co-option I showed you before, but also you're by serial duplication, which is a relatively easy thing to do genetically and evolutionarily. By serial duplication, you end up with a more complex genetic repertoire to build bodies. And I think that's really, really, really important. So what I've talked about so far in terms of our list of innovation and evolution is repurposing, change in function, how structures can have multiple functions, and it's important to think about locomotion that way. Co-option and redeployment, we see that at the level of genetics, that you can co-opt an existing generative mechanism, you co-opt it and redeploy it. You can take a generative mechanism and duplicate it and modify it, which not only increases redundancy in the system, but it also gives fodder for new evolutionary change. This is all fuel for evolutionary change, right? And this is how like embodiment evolves. These are the basics of that. And what I want to do is I want to end with about 5 to 10 slides on the relationship between sensation and morphological evolution and feeding. And I think it'll take you to what I hope is a very surprising place. So I want to end with a surprise. And this is something that's coming out of my lab. It's unpublished, but it's kind of in the process. All right, so teeth. We're going to talk about teeth. Feeding. Think about, locomotion has been the first part, but now we're going to look at teeth. So if you look at teeth, all kinds of different teeth out there. You see the human, you see a shark, you see piranha, we see a dinosaurs, you see a smiling zebra. All right, so this is what we're going to do. We're going to talk about teeth, but to do this, we have to go way back in time. We have to go back about 480 million years now. We're taking even deeper than limbs. We have to go to the earliest vertebrates. Now, the earliest vertebrates are jawless fish. All right, so what you have here is a phylogenetic tree, pretty much of vertebrates. If you look on the bottom, what you see cyclostomes, see at the bottom of the tree, then the clear part, those are jawless fish, just alive today. That's like a lamprey or a hagfish. They have no fins, they have no jaws, they're pretty primitive. If you look at the green part, those are fossils. Those are fossil jawless fish. There are a variety of fossil jawless fish. They're known from rocks over 450 million years old. Then as you go higher in the tree to the blue, you get into the sharks. And then you get into all the different kinds of fish, including us. I'm calling us fish here. See the tetrapods in the center there, all kinds of different fish. The key point is here, to understand the origin of teeth, we have to go to the green part of the tree, the early vertebrates, the earliest vertebrates. And what you see on the right is what their bodies look like. It's really kind of different from anything on the planet today. They have an exoskeleton. So a lot of their body is covered with bony plates. And you can see them outlined here. Their heads have these, typically often have these big shields, like almost like a hamburger, but shaped like a hamburger, but they're big fat plates of bone. So they have all kinds of like plates on the outside of their body. All right, so that's key. Keep that in mind. We're looking at jawless fish at the base of the tree. And what these jawless fish have are bony plates on the outside of the body. Now, what does this have to do with anything? Well, here's what happens. So let's take a peek at these plates on the outside of the body. And paleontologists have known this for a little while. So this has been sort of a textbook story for a little while. If you look at the plates, they're actually composed of lots of bit of tesserae, like little tiles, okay? Hundreds of them. And if you look to see the box in the center there, that's of one of those little tiles. And that tile has a very characteristic structure. It's called an odontode. And the reason for that it's called that is because when you look at it, it has an outer layer shown in yellow of enamel, an inner layer composed of sort of a tubular kind of bone, which is dentine, and it has a pulp in the middle. So what you have is enamel, dentine, and pulp. Does that sound familiar? It should, because it's very similar, almost identical to teeth. These are teeth which are in the inside of our body, which you have enamel, dentine, and a pulp cavity. And these are all attached to bone. So what you see on the left is an odontode from the outside of the body of an early vertebrate. And what you're seeing on the right are in an incisor and molariform tooth of a human, right? But yeah, I could give you anything else, any other taxon, but that's good enough. You get it, right? Well, what's interesting here is paleontologists for a long time have held the idea that of what's called the outside-in hypothesis for the origin of teeth. That is, teeth originally arose as armor on the outside of the body in these odontode-like structures, and ultimately over time got incorporated in the epithelium on the inside of the mouth. So teeth originally evolved on the outside of the body as parts of the armor, and now are functioning and feeding. Okay, so that's there's a lot of data behind that. And it's surprising, but true. Again, nothing ever begins when you think it does. But it gets even stranger. So what we've been doing is looking at odontodes from some of these really early vertebrates and using high energy synchrotron scanning, like a really advanced photon source to get inside. And you can scan inside, and here's one of those odontodes. This is of one of those species. And you can see there's thick enamel on the outside, but on the inside, you see all these different tubules. See a little tubule space there on the right? That's got microdentine tubules is labeled. A lot of these tubules. It's a characteristic dentine structure. So we looked at them, and they're very characteristic. And you can see in the earliest vertebrates, here's an early vertebrate, you can see you have tubules which define, they go from a pulp cavity. These tubules extend outwards to the outside of the animal in these odontodes. And you see different flavors of them. So what we decided to do was say, well, how unique is this system? What if we looked at like fossil arthropods, not vertebrates with bone? What if we looked at another kind of creature, cuticle? I mean, what's going on here? So we did that. And so we looked at fossil arthropods from the Cambrian. And guess what we found? Tubules. All right, now if you saw this on the left there, you would think that was a odontode, a vertebrate odontode, but this isn't a fossil arthropod, like a trilobite kind of animal. It looks like a trilobite. And it has those. And if you look at a horseshoe crab, if you look at a crab today, if you look at the cuticle inside the armor, it's not made of bone, right? It's a mucopolysaccharide. They have these tubule structures as well. So odontodes are very similar to the structures in non-vertebrate animals in their cuticle. And so in odontodes are in bone, but they have a similar structure in the cuticle of crabs and stuff like that. All right, so what's going on? Well, it turns out these are sensory structures. So these structures are connected to what's called sensillae. They function in sensation, mechanoreception, chemoreception. What they are a series of tubules that connect to the outside. Some of them have like connect to a little hair, which is a sensory structure. So we decided to look. Let's look at, I mean, there are creatures that have odontodes, vertebrates. So here you have a similarity between vertebrate odontodes in skeleton, the outside of their body, and the sensory organs of arthropods, right? Crabs and bugs and fossil versions of that. It's pretty crazy. So our teeth are very similar to these. So we decided there are creatures that have odontodes on the outside of their body that are alive today. So there's some catfish, there's some sharks, and so forth. So we asked the question, are they innervated? So here's a catfish, and then you can see in the outside of the body it has an armor, kind of like those fossil creatures. And you can see the head on the left, you can see the body. And those are all odontodes. Those all have the same structure I showed you in the fossils with a little bit of enamel, a tubule system, and so forth. And guess what? When you look at teeth, you know this when you go to the dentist, teeth are highly sensory, right? They have a sensory component to them, which is necessary for chewing. There's a, somatosensory proprioceptive feedback to get the right bite. Close your mouth and you always close in the right place, right? Well, there's a lot of sensation that happens there. People who have lots of implants tend to break their teeth more than people who don't. And we know we're very sensitive. Anytime you've been to the dentist, you're well aware of how sensory teeth are. And indeed, the cells that make teeth, the odontoblasts, actually partially sensory cells themselves. So it's really interesting. So there's this tie between teeth and sensation. But if we look at odontodes on the outside of the body, whether they're in fins or heads, what we find is they have sensory innervation as well. So odontodes that are on the outside of the body in fossil creatures are similar to the sensory structures of invertebrates. The odontodes that we see in living vertebrates today, whether they're teeth or scales, are all innervated with sensory innervation. What that means is that our teeth originally arose on the outside of the body as sensory organs for mechanosensation. And then over time, they were recruited and co-opted and redeployed in the inside of the body, in the mouth, to form a dentition. So every time you go to a painless dentist, you can thank fish living in aquatic ecosystems 470 million years ago, 480 million years ago, adapting the sensory system in new ways. So here what you're seeing is that what you're finding is the locus for selection and the directionality of evolution changes with each redeployment. That is, you had these structures, evolving in a sensory milieu, right? In the Ordovician, maybe even the late Cambrian, right? But probably Ordovician, which is 480 million years ago. Then as they redeployed in the inside of the mouth, they retain their sensory functions, but then they took on a whole new set of functions for mastication. So it's basically evolution by repurposing. So to sort of close and to talk about innovation and evolution, and hopefully this is fodder for thinking about embodiment and how it evolves, is innovation can happen. The toolkit for evolutionary change really is, repurposing. Again, in the Lillian-Hellman context, nothing ever begins when you think it does. Using old organs in new ways, and thinking of, well, maybe there's not just a single use for an organ or a function. Maybe it can be deployed in diverse contexts as an environment that those animals lived in. So we tend to simplify as humans in a heuristic sort of way, right? That, single function, single directionality of evolution. That's not the case at all. Evolution goes back and forth all the time. And ancient organs can be used in new ways. Co-option and redeployment, the generative mechanisms that form organs that can arise in one context to form one body part, like the body plan of a fly, and then can be redeployed to make appendages like fins and limbs and genitalia and so forth. So cooption redeployment. Big time fuel for evolution is duplication and divergence. Why? I mean, duplication is a real quick way that evolution uses to make a whole new system, like duplicate a genetic network, duplicate a set of genes. And there's really simple ways to yield massive genetic change is through gene duplication. I didn't talk about it today, but another way that that innovation in evolution is mergers. Creatures come together to make something new. So you can have viruses going between animals that share genetic information. I mean, 9% of our genomes are ancient viruses that invaded our genome years ago. And some of those actually are proteins we use today in pregnancy and immune response and so forth. So the origin for some of our physiological traits is actually from viral exchange. Endosymbiosis, where creatures can merge in certain ways. And the other big thing in evolution, this is kind of where I want to close, is timing is everything. That you can have the right invention, but it's really the opportunity that's essential. It's you know that when you think of technology, right? I mean, you need the context, you need the distribution system, you need the software. You can have the best hardware in the world, but the software or the apps, it's not going to work. So timing is everything. And when you think about change, and when you think about repurposing, cooption, redeployment, duplication, mergers, the way an innovation gets traction really depends on what the state of the system is at that moment, the external system and the balance between the external system and the internal one. Okay, on that I'll close. I just want to end with some thank yous. Tons of people behind a lot of the work I talked today, tons of funders from the US government to private foundations, close colleagues, some of whom are no longer with us, postdocs shown in red. The Inuit and indigenous communities have been really important for our work up in the polar north. Want to thank them. My current lab, consisting of undergraduates and graduate students, have been really pivotal. And and just thank all the field crews. I mean, you don't do this alone. I mean, there's like 50 people behind finding Tiktaalik. So, and I owe thanks to all of them. With that, I'll stop and thank you very much.
Thank you so much, Neil. That was a fantastic talk that really gave us a lot of insights. And I see we've got quite a few questions. And so that's hopefully we've got you for a little bit more just to
you got me. I'm hearing. No, you got me. I'm online.
So just to check I've understood correctly. So if we think of animals as being, from an engineering perspective, we've got morphology, we've got materials, we've got sensory aspects. And so what you're saying is the timescales over which they evolve is different, or you may have a period of morphological adaptation when the materials have already developed, and then you may have sensory adaptation. And the timescales and the evolution that these happen is very different and driven by different kind of shifts or environmental changes.
Exactly. Yeah. And you might have a change in, a structure that really sets the path of sensory evolution and functional evolution off in an entirely new direction, and so what you're seeing is you can have different pathways of evolution because structures, functions, and the way you sense the environment can shift in different time frames, and each of those can yield a different direction, if you will.
I was wondering, what can these also tell us? So obviously you're looking at kind of physical structures, but what can we extract? What can that tell us about the cognitive capabilities and the evolution of or can you make any inference about the evolution of the cognitive capabilities or the plasticity of the cognitive capabilities from the kind of physical evolution?
Yeah, I mean, you think about the environment that a land-living animal exists in versus one a fish lives in. The sensory environment is very different, right? All of a sudden, you're in a much more visual world. Creatures that live in on land, it's a vision world more than the mechanosensory.
Yep.
Chemosensory. I mean, they're still there, of course, obviously, but I mean, but vision becomes the thing, right? And you go from where a more mechanosensory world to a vision-based world. And when you think about and olfaction also begins to become much more important. And so you see the olfactory genes increasing a lot. So you're changing the sensory environment. But the world is, and when you get to the terrestrial world, is much more, is textured in a different way, you know? And so what you start to see is, and again, I can't infer, we don't have the brains, but I can infer from, what these animals are doing. Their terminal appendages are much more complex. Their sensory environment with the visual world is much more complex. More of the brain becomes dedicated to sensation, particularly because vision is a very expensive sense, right? It's more so than some of these others, and expensive in a lot of ways. And so, I mean, I think with that has to be new ways of cognition, right? Because you're changing the sensory environment and the ways that animal have to perceive that world, and the ways that they move about it. So I think, the cognitive ability and the cognitive substrate that's necessary to even just do those basic functions has to increase pretty dramatically once you make the transition to land. So I think that's right. I mean, and you happen to do see in a gross way a change in brain size, body size relationships during this transition too. So that's probably a little bit about it. And so indirectly getting to the thing of cognition, because I don't know directly, but certainly you're getting more brain to body size.
Thank you. Very interesting. And one question on the chat that I think is very relevant is talking about, okay, very interesting notion that there's this blueprint that can be adapted and morphed into whatever creature fits the environment. But how does this blueprint get defined in the first place? And I think this is very interesting from an engineering perspective. This is this question of encoding and DNA, and do we need an encoding to be able to evolve? And how does that create? So I wonder if you have any thoughts regarding that.
Yeah, so there's a couple things that are really important here. When you think about call it the blueprint, I like to think of it as the recipe, but okay, whatever. But I mean, I think the more we look, the more we find as developmental biologists and geneticists, biochemists, is that self-assembly under the right conditions becomes a very powerful force. So if you have the right sort of physical and chemical external conditions, that is, take an embryo, cells can come together in very complex patterns, just truly by self-assembly. If you do the right, and physics and chemistry can take over as the cells interact to give you highly complicated patterns. So I think when you think of, the earliest, I'll call them blueprints, whatever you want to call them, they certainly are not going to be identical to the derived ones. They'd be much more simple in some ways, but self-assembly is a big part of that. But also I think, you can have relatively simple structures and functions that can do a variety of different things. And then that sets them up to go for that to be co-opted in it and take it into a new direction to get more complicated in different ways. What I'm saying is you begin simpler. I mean, so the blueprint is not identical when you go all the way back. You have you have fewer duplications, fewer parts of the network system, fewer, so there's a simplicity to it, but you have enough of that blueprint that it can, take them in different directions. But no, I think self-assembly is actually, the more we look, the more we find. So, and I'm thinking of that in terms of the developmental world. People are now like building embryos in dishes. What they'll do is take stem cells, and they'll just basically take them, mix them up, put them in a dish, and they'll put them in different settings, like put a little barrier around them, so they have to develop like in an egg or something like that. And what they find is these things can get incredibly complicated patterns that are very similar to embryos, which is shocking. They can get the 3 germ layers. There's already information there, and so so self-assembly, I think, is a big system. That doesn't answer the question entirely.
But it's a very interesting answer.
Yeah.
No, I think it's a really interesting question. So perhaps